I really appreciate everyone coming out today to show their support for our work that we've done this summer. I'm going to tell you a little bit about an ongoing project that we're working on in the Holland Lab that looks at using RNA vaccines to treat cancer. This summer, we focused on targeting gene fusion positive cancers. I was lucky enough to be placed in the Holland Lab at Fred Hutchinson, and in the Holland Lab, we study brain tumors with a focus on glioblastoma and aggressive recurrent meningiomas. Our research focuses on building mouse models of human cancer that can be used for preclinical testing of therapeutics, as well as analyzing the function of the drivers of cancer, like gene fusions and aberrant splice forms that are found in cancers. The foundation of our model is the RCAS-TVA system that allows us to induce in situ tumors, which develop in 1 to 6 months, depending on the model. So most of us are pretty familiar at this point with how RNA vaccines work, thanks to COVID, and ours works pretty much the same way. The idea is that we're going to harness the immune system to attack intracellular antigens. The difference is that instead of attacking a pathogen, we're asking the immune system to target something that comes from within. And the issue here is that cancer is essentially self. If we tell the immune system to attack self, it becomes highly problematic. So the solution is that we need to target a driver of cancer that isn't found in the body outside of cancer. So in our lab, we've identified fusion genes as a target. They're present in all forms of neoplasia. They contribute to tumor formation due to their tendency to produce these excessively active abnormal proteins. They're commonly found as oncogenes, and they're most prevalent in pediatric cancers. And the way that they work is they constitutively activate oncogenic signaling. So you can see on the right here, sort of an illustration of how it forms. You get these rearrangements of chromosomal or somatic chromosomal rearrangement, which causes the gene fusion, which leads to these fused proteins. Okay, so we have just begun this really incredible discovery study that's going to address the question, can we show immunoprophylactic effects or treatment effects vaccinating against these drivers of cancer? So our goal here is to get meaningful reduction or prevention of tumor growth. So our hypothesis for this very first stage is that RNA vaccines can prevent tumor growth by targeting these oncogenic gene fusions. We've chosen these specific fusions that are listed up here because we work with them all the time, and we know they're very reliable, and we know they're aggressive. So it's going to lead to a really good readout. If this system that we're developing works, this will be the best model in which to prove that. So because this is a discovery study, much of our work this summer has been centered around sort of developing the method and the protocols. This illustrates how we are testing our system. First, we generated this specific gene profile that's going to allow us to run the system effectively. We ran a toxicity study to make sure that the vaccine was safe. We did a tumor growth control study where we induced tumor growth on each side. This is a really critical step because later on we're going to be showing evidence that's basically nothing. So by doing this, we can show that in the absence of the vaccine, we're still able to grow tumors. And then we're running an assay currently that's going to allow us to show that our vaccination process is effective. Okay, so this is our experimental plan. We have done the first 2 rounds of vaccinations. On Monday, I'm going right back to work, and I'm doing RCAS-TVA flank injections. We want to focus on the flank first because we go straight to the brain. There's an issue of the blood-brain barrier possibly having an effect. So we want to show this works in a flank tumor first, and then move on to brain tumors. And then after that, we're going to do the 3rd round of vaccinations, and then we're going to be evaluating the immune response. Okay, so what we're going to see, if it doesn't work, we're not going to see any difference between our control and vaccinated mouse. We're going to go back and reevaluate the design if that's the case. It could also possibly work initially, but then get a rebound in the tumor growth. At that point, we need to start looking at escape mechanisms. If it does work, ideally, we're going to see nothing. The tumor's not going to form, and then we'll be able to do tests and see if we can show an immune response and an impact on the tumor growth. And at that point, we then get to pivot and get a mouse that already has an established tumor and see if we can vaccinate away the tumor. This is really the most exciting step for me because this is how we would treat humans. This is how we treat an actual patient. We're not going to pre-vaccinate people against these tumors. We need to be able to treat a patient. So when we finish our work with gene fusions, we're going to move on to our next target, which is this T1 isoform. It's a really exciting target because this isoform, unlike the gene fusions which are found in these really rare pediatric cancers, we believe that T1 is a driver of many, if not all, cancers. So right now we have a manufacturer working on the vaccine. When it arrives, we're going to be able to hit the ground running because we've already done all of this work through the gene fusions. I would like to thank all of the many wonderful people that have played a role in my summer of discovery. All of my many mentors at the Hutch, including my PI, Dr. Eric Holland, the incredible scientists in my lab, Frank, Debbie, Dennis, Zach, Maddie, and James, and everybody else. This has been a really incredible experience for me. I told Dr. Holland the first time I met him that I felt called to the work. And after this summer, I have realized that this is what brings me purpose. And for that, I am truly grateful to everybody in this room, everybody on Zoom. Thank you so much. I want to thank Drs. Ellen Bogan and McDonald, Jim Pridgeon, Julie, and Sylvia, the selection committee, and all of our amazing donors. I'd also like to thank my incredible cohort of students. To quote a very wise woman that I know, this summer with all of you has been a 12 out of 10. Yeah. And then I have one last thank you to my dad. His experience with neurological surgery taught me that you can face this with a sense of humor and curiosity rather than fear. And thank you so much.